Assalamualaikum warahmatullahi wabarakatuh everything. Today we will discuss about population genetic. We are from Group Tonto. My name is Muhammad Daniel bin Mustafa Kamar.
And my name is Syamil Wafi.
My name is Muhammad Suyullah bin Muhammad Nur.
My name is Muhammad Aiman.
And my name is Muhammad Amirul bin Mulyan. So let's start the discussion about population genetic. First, we're gonna define what is population genetic. So, population genetic is a study of genetic variation within population, and it involves examination and modeling of change in the frequencies of genes and alleles in population. So, genes found within population is also known as polymorphic. So, the factors of population genetic including population size, mutation, genetic drift, natural selection, environmental diversity, migration, and non-random mating patterns. So, an example of population genetic is founder effect, which is loss of genetic variation occurs when new population established by very small number individuals from a larger population. For example, of founder effect is African population of Dutch settler in South Africa descended mainly from a new colony. So, what about you, Eman?
Okay, I will talk about the importance of population genetic. Well, firstly, we need a large amount of variation within a population for the survival of the species. When having a large levels of variation, we are more likely to have traits within the species to overcome any environmental changes. Some species have a very good variation, but if something comes along to jeopardize their habitat, they have a very high chance of going extinct because they have no way of avoiding or removing the threat. Further, the variation within a species allows for development of new trait combinations which can allow a species to improve upon itself over time, hence allowing for better eyesight, speed, and strength. And finally, being able to monitor the genetics of population allows the scientists to monitor the variation between the species, which is noted above, can aid in seeing the development of a species or even change one of the species into another, as well as the potential robustness of the species when faced with any environmental changes. Okay, Sufi.
So now I will talk about Hardy-Weinberg. So what is Hardy-Weinberg? So Hardy-Weinberg explains why a length frequency does not vary throughout the generations. From generation to generation, it's still the same. And it also describes mathematical relationship between allele frequency and genotype frequency. Third, it allows prediction of genotype frequency from allele frequency. And random mating is important in the principle of Hardy-Weinberg. Population mates randomly, which will produce diploid genotypes by random haploid eggs or sperms. So Hardy-Weinberg applies when the large populations there are no genetic drift or founder effect in the large population. Secondly, it must be random mating. Third, the mutation rate must be normal, so the little alleles are replaced by new mutation. And lastly, no selection of genotypes, no migration, no gene flow into or out of population. So Wafi, you want to say anything?
I want to say a lot of things actually. I want to say about factors that affecting allele frequency. Just now, our friends here said that factors affecting population genetic. We will discuss about the factors affecting allele frequencies, which are genetic isolation, migration, mutation, and also artificial selection. Okay, for genetic isolation, it is a separation of interbreeding population of animal that into two or more group of other animal that do not exchange gene. Okay, that's general isolation, genetic isolation. Then for migration, it tells that allele can flow from one population to another when the animal migrate and begin to interbreed into a new location. Okay, for the mutation are the important source of allele variation because it provide the new gene, it provide raw material for gene to evolution of gene. Mutation always occur continually that can increase frequency over many generations. Okay, that's for mutation. And then lastly about artificial selection. Artificial selection is involve man's effort to change the genetic constitution of animal population by choosing the parents of the next generation. We human do that by the selecting for desired characteristic of gene through many generations. Okay, that's all for factors affecting gene allele frequencies. So, for Daniel,
Thank you, Syamil Wafi. I want to talk about several terminology that we must know in this topic. Okay, first, this is frequency. Frequency that a genetic disorder is observed in a population when in 150,000 people. Mutation frequency. Frequency that a mutation occurs in the DNA. Number of mutation per locus per gamete per generation. Okay, next, polymorphism. The occurrence of two or more alleles at a specific locus in frequency greater that can be explained by mutation alone. Common in non-coding region of DNA, does not cause genetic disorder but can be used as a genetic marker for a gene. Okay. Next, single nucleotide polymorphism. Silent mutation that accumulate in the genome.
oh,
Okay.
That's right.
Restriction, fragment, length, polymorphism. Polymorphism that either creates or destroy a restriction enzyme site abundant throughout the human genome used in gene linkage or gene mapping studies. Okay. That's all from me. From us. Thank you.
Thank you.
Bye.
see you soon.
How's that going?